Bioenergetics of Exercise and Training by Joel Kramer, Ph.D., and CSCS with distinction, and NSCA, CPT with distinction.
This presentation is entitled Bioenergetics of Exercise and Training. To start this lecture, we thought we would cover some basic definitions that will be used throughout the lecture interchangeably, so understanding these concepts will be important throughout the remaining lecture. Energy is the first basic definition. Energy is defined as the ability to do work. Work is defined mathematically as force times distance. So initially, if you think of work as a mechanical aspect, where if doing a 1 repetition maximum bench press or squat, work is force times distance, it's easy to calculate the amount of work done. But work from a metabolic sense can occur on the cellular level, even in a mechanical sense can occur on the cellular level as a cross bridge fires and a muscle can contract. That's also mechanical work. Metabolic work can occur when making larger molecules from smaller molecules. And so in either case, energy is necessary to do that. Bioenergetics is kind of a large term that encompasses everything, the flow of energy in a biological system, which we defined earlier. Catabolism and anabolism are two opposing terms. Catabolism is the breakdown of larger molecules into smaller molecules. An example of this is in glycolysis, where we break down blood glucose into 2,3-carbon pyruvate molecules. Anabolism, on the other hand, is making or synthesizing larger molecules from smaller ones. An example of this would be making a protein, polypeptide, from smaller amino acid residues. In a bigger picture, that polypeptide or protein can be used at the muscle cellular level to replenish or fix microtrauma in a muscle and make the muscle larger, a process known as hypertrophy. Like anabolism and catabolism, or in this case catabolism and anabolism, exergonic reactions and endergonic reactions are similar opposing terms. Exergonic reactions refer to energy-releasing reactions. Generally, they are catabolic. Endergonic reactions are energy-consuming reactions, and generally, they are anabolic. An example of exergonic reactions would be when we break down muscle glycogen or blood glucose during catabolism. The end process is a release of energy, or at least a release of energy in the storage form of ATP. Endergonic reactions, and examples of those, such as making proteins or polypeptides, are energy consuming and they require the use of ATP. Metabolism is a term that encompasses pretty much everything, and that is the sum of all catabolic and anabolic reactions, or exergonic and endergonic reactions in a biological system. So overall, the term metabolism and bioenergetics are very similar and can be used interchangeably. The last definition that we'll cover, as far as the basic definitions go, is adenosine triphosphate, or ATP. ATP is the intermediate molecule that allows the transfer of energy from exergonic to endergonic reactions. We will use the term ATP throughout this lecture many times, so it's important to be comfortable with the term. ATP is the smallest usable form of stored energy in the body. Understanding the structure, the basic structure of ATP may also be helpful. ATP is made up of adenosine and triphosphate, as the name suggests. Adenosine is made up of a 6-amino purine adenine and a 5-carbon carbohydrate called ribose. Those two link together and form adenosine. And the triphosphate group is simply 3 inorganic phosphates linked together that are attached to the ribose pentose ring. It's those 3 phosphates that contain the stored energy, the bonds between those phosphates, that create the stored energy that ATP is known for. Cleaving the terminal phosphate, and which results in an ADP, or adenosine diphosphate, and a free inorganic phosphate, releases the energy necessary to do a great deal of work, some of which you've covered in some of the other lectures, one being the cross-bridge recycling mechanism, which we'll discuss quite a bit, the cleavage of ATP, otherwise known as the hydrolysis of ATP, supplies the energy necessary to fire one cross-bridge. Figure 1 shows a large general concept of metabolism. Bioenergetics, or metabolism, the flow of energy within a biological system, is depicted in Figure 1. Where large molecules are broken down into smaller molecules, this process, like we termed before, known as catabolic or exergonic, creates the energy necessary to resynthesize ATP from ADP, again, adenosine diphosphate. That ATP is stored energy and provides the energy for smaller molecules to be built into larger molecules, again, that reaction known as anabolic or endergonic in nature. ATP is also used for mechanical work, as we described before, but Figure 1 should provide a general holistic view of metabolism that we'll talk about throughout this lecture. It's important to keep this big picture in mind because as we go through the individual pathways of glycolysis and the Krebs cycle and the electron transport chain, this big picture is really what we're trying to get at, and that is the flow of energy in a biological system. When we discuss the replenishment of ATP, obviously we're aware that we use a lot of ATP for the work that we do. When we replenish ATP, there are three basic systems that do this in the human body: the phosphagen system, the glycolytic system, and the oxidative system. They progress from the lowest capacity to the highest capacity, and then from the most rapid replenishment of ATP to the slowest replenishment of ATP. And we describe this a little bit later on in this lecture. But it's important to understand that the phosphagen system, being a very rapid replenishment of ATP, has the lowest capacity, whereas the oxidative system is a slower replenishment of ATP but has the largest capacity for ATP replenishment. The phosphagen system and the glycolytic system can be grouped on a couple different issues. First, they both occur in the sarcoplasm, whereas the oxidative system actually occurs in the mitochondria. So there's two different locations within the cell that these systems occur at. Second, the phosphagen system and the glycolytic system are defined as anaerobic energy systems that can occur without oxygen in the cellular environment. Now, when we say there's no oxygen in the cellular environment, that's not entirely technically correct. There's very little oxygen and not sufficient enough for oxidative metabolism to occur. Oxidative system or oxidative metabolism that occurs in the mitochondria must occur in the presence of oxygen. Therefore, there must be sufficient oxygen in the cell for oxidative metabolism to occur. It's also important to recognize that even though there are three separate energy systems, they are not exclusive. All energy systems are active at any given time. Take, for example, two scenarios. One where you may be sitting writing right now, and another where you might be exercising. All energy systems are active during those times. While you're sitting and writing or near rest conditions, the primary energy provider is likely to be oxidative metabolism. However, the phosphagen system and anaerobic metabolism may be contributing during writing muscular actions of the fingers. Whereas during exercise, all three energy systems are also active. And the extent of contribution of those energy systems depends primarily on the intensity or power output or work rate of the exercise, and secondarily on the duration of the exercise. When we talk about, a little bit later in this lecture, when we talk about which energy system is the primary contributor, or this energy system may contribute for a certain duration of time, the duration of time is really dictated by the intensity of the exercise or the intensity of the activity. The lower the intensity of the activity, the longer the duration can be. The higher the intensity, the shorter duration. Obviously, if we exercise at extremely high intensities, we fatigue very rapidly, and that fatigue will dictate the duration of the activity. But when we talk about power output or intensity of the activity, we talk about work or force times distance, mathematically, divided by time. So work divided by time enters in the concept of time being a part of power output. And when time is a factor, intensity is going to be a larger issue to dictate the duration of the exercise, and particularly dictating which energy system is the primary contributor to the activity. And as we go through this, we'll use examples of exercise to show what particular energy system is utilizing what particular is utilized during and exercise. We'll begin by talking about the phosphagen system. The primary functions of the phosphagen system are, one, to provide ATP for high-intensity activities, such as sprinting or weight training. This energy system can last approximately, or can be the primary energy contributor for approximately 0 to 6 seconds, and can last up to 20 or 30 seconds of the activity. There are other names for this system that include the ATP-PC system, or adenosine triphosphate phosphocreatine system. The phosphocreatine is one of the substrates involved in the reaction that is the crux of the phosphagen system, which we'll see on the next couple slides. Regardless of the intensity of the exercise, all exercise starts by using the phosphagen system. So when we start jogging on a treadmill, for example, the very first part of that, the first few seconds of that jogging activity, the primary energy provider for that initial part of the muscle actions involved in the jogging exercise is the phosphagen system, even though it may be a submaximal activity. The first reaction in the phosphagen system, or in figure 2, is actually the breakdown or hydrolysis of ATP. Obviously, if we need to replenish ATP, we first have to break it down. And when ATP is broken down via myosin ATPase in the figure, it results in the products of ADP, inorganic phosphate, and energy. To replenish that ATP with the phosphocreatine system, or the phosphagen system, ADP, or adenosine diphosphate, one of the products of the hydrolysis of ATP, is combined with creatine phosphate and catalyzed by creatine kinase enzyme to create one ATP molecule and creatine. This reaction is really the basis of the phosphagen system and is utilized to replenish ATP very rapidly. The second equation in figure 2 is the myokinase reaction. The myokinase reaction involves taking two ADP molecules, bringing them together with the myokinase enzyme, which results in ATP, adenosine triphosphate, and adenosine monophosphate. If you take two ADP molecules, each with two phosphate groups, take one phosphate from one of them and add it to the other one, we can get an ATP and an AMP molecule as a result. If we go back to the first equation, the hydrolysis of ATP, ATP can be broken down in several different locations in the muscle. The location will determine what enzyme is used to break down ATP. For example, in figure 2, it shows myosin ATPase hydrolyzing ATP, the ATP molecule, for the energy release. But other enzymes can also break down ATP. For example, sodium-potassium ATPase. Myosin ATPase is used to break down ATP at the level of the myosin head during cross-bridge recycling. Sodium-potassium ATPase breaks down ATP for the energy necessary to pump sodium out of the cell and potassium into the cell, hence the sodium-potassium pumps. Sodium-potassium pumps require energy. The third one that we'll discuss is calcium ATPase. Calcium ATPase is necessary to pump calcium back into the sarcoplasmic reticulum. If you recall the process of excitation-contraction coupling, calcium is necessary for the formation of the actin-myosin complex, or the formation of actin and myosin. During that process, calcium has to be present and is released from the sarcoplasmic reticulum. But in order to get it back into the sarcoplasmic reticulum, it takes energy, and it has to be actively pumped back in. So to break down the ATP for that energy, calcium ATPase is used to hydrolyze ATP. So as a summary of the phosphagen system provides a very rapid ATP resynthesis rate. It can resynthesize ATP that has been broken down very rapidly. And it's efficient. There's only a couple of reactions involved. Once ATP is broken down, it can be resynthesized with the creatine kinase reaction, or it can be resynthesized with the myokinase reaction. That's only one reaction involved in the resynthesis of ATP. In contrast to the glycolytic system, where there's 12 to 14 reactions involved for the synthesis of 4 ATP molecules. So efficiency is, and the speed of regeneration is a benefit of the phosphagen system. But the trade-off is a very low capacity. creatine phosphate can be depleted from the muscle or almost virtually eliminated from the muscle during high-intensity exercise. The next system that we're going to discuss is the glycolytic system. The primary functions of the glycolytic system are to break down carbohydrates. The two forms of carbohydrates that we're going to break down are blood glucose and muscle glycogen. The purpose of this system is to produce ATP in the cytoplasm or the sarcoplasm of the muscle cell. When transitioning from the beginning of exercise, which we know occurs, the primary energy provider for that is the phosphagen system, the glycolytic system starts to take over after approximately 30 seconds when the phosphagen system can no longer provide the necessary energy, if the intensity is low enough to last that long. So the process of the glycolytic system can provide energy for moderate to high intensities for activities lasting between 30 seconds and 2 to 3 minutes long. This occurs in an anaerobic cellular environment. We defined anaerobic earlier. In your notes and in the slides, we use the term hypoxic cellular environment also. Hypoxic is another term used to define anaerobic or reduced amounts of oxygen, hypoxic cellular environment. Next, we're going to discuss Figure 3, which is an overall summary and a basic summary of the glycolytic system. Figure 3 has a lot of reactions in it, and it's important to remember one thing before we start talking about this pathway, and that is the reactions occur in steps. First there's a reactant, then there's an enzyme, and then there's a product. The reactant is the substrate, the enzyme must be present to catalyze the reaction, and the product is the end product of that particular reaction. For example, the hydrolysis of ATP. ATP is the reactant, myosin ATPase is the enzyme, and then the products are ADP, adenosine diphosphate, a free inorganic phosphate, and energy. So the direction of that particular reaction occurs in that way. Figure 3, although it looks intimidating, is really a basic overview of glycolysis. There's about 5 issues that we need to discuss regarding glycolysis and the energy being produced within it. The first is the creation of glucose 6-phosphate, or G6P. It's located in the center upper portion of Figure 3, and it can come from 2 sources. The first is blood glucose, and the second is muscle glycogen. If it comes from blood glucose, it has to be phosphorylated to maintain or stay within the cell. Blood glucose comes in and out of the cell, and in order for it to remain in the cell, it must be phosphorylated. Unfortunately, that phosphorylation costs us 1 ATP. Whereas from muscle glycogen, the production of glucose 6-phosphate does not cost an ATP. Muscle glycogen is just stored blood glucose. So when blood glucose links together and stores within the muscle, it's called muscle glycogen. And since muscle glycogen is already in the muscle, it does not need to be phosphorylated. So the process of the formation of glucose 6-phosphate, or G6P, from blood glucose occurs with the presence of the enzyme hexokinase. And again, it costs us 1 ATP. So although it may seem initially counterproductive that the glycolysis pathway, which is intended to create ATP or synthesize ATP, the first step, or the priming reactions, which we'll discuss in a moment, are actually costing ATP, which will reduce our net ATP production. The second main issue within the glycolytic pathways is the phosphofructokinase reaction, or PFK. PFK is an enzyme that is important for 2 reasons. First, again, it costs 1 ATP. Regardless of whether it comes from blood glucose or muscle glycogen, that ATP is used to form fructose 1,6-bisphosphate. So that is an example of an anabolic endergonic reaction. The second important thing is that PFK is the rate-limiting enzyme. PFK is a large enzyme that has many binding sites for inhibition. For example, hydrogen ions that accumulate during fatigue can bind to PFK or react with PFK to slow glycolysis down. Other things that can bind with PFK are lactate. Lactate can also slow the process of glycolysis down. Therefore, phosphofructokinase, or PFK, is sensitive to the internal environment of the cell and can be limited by a number of different factors. The third key issue to discuss is that all the reactions leading up to fructose 1,6-bisphosphate, which you can see in the center of figure 3, have been priming reactions. They've been priming because it's preparing the 6-carbon chain, the 6-carbon carbohydrate chain, to be split into 2 3-carbon chains. The 2 3-carbon chains are glyceraldehyde 3-phosphate, located toward the center of figure 3. If we start with blood glucose, we've already used 2 ATP, so we're already 2 ATP in the hole. If we started with muscle glycogen, we are 1 ATP down. And again, although this seems counterproductive, these priming reactions have prepared the 6-carbon chain to be split into 2 3-carbon chains, which actually result in a net positive production of ATP. The fourth issue is the energy, or the ATP, that is produced as a result of glycolysis, which is the whole purpose of the pathway. Since we're already down 2 as a result of the metabolism or priming of blood glucose, and down 1 as a result of the priming of muscle glycogen, we now are prepared to actually produce ATP as a result of the pathway. The first potential energy source that is produced as a result of glycolysis is the reduction of NAD. There's 2 issues I want to talk about NAD and reduction. The first is NAD. NAD stands for nicotinamide adenine dinucleotide. NAD is reduced and carries or transports a proton. Now, go back to your oxidation reduction reactions in basic chemistry. Oxidation and reduction refer to the relative proportion of oxygen within a molecule. If you reduce a molecule, you reduce the relative proportion of oxygen within that molecule. Therefore, when we add a hydrogen to NAD, we reduce the overall proportion of oxygen. So NAD is reduced to NADH. In the step after the reduction of NADH, we actually get substrate level phosphorylation. Here's another term that we'll define briefly. Substrate level phosphorylation is the production or synthesis of ATP as a result of a reaction directly involved in the glycolytic or Krebs cycle pathway, as opposed to oxidative phosphorylation, which is the production of ATP by the oxidation of NADH. We'll talk about oxidative phosphorylation when we talk about the electron transport chain. But as far as substrate level phosphorylation, we get our first ATP production after the reduction of NADH. The second substrate level phosphorylation and production of ATP occurs when pyruvate is created and ADP is converted to ATP. Now notice that there are 2 columns in figure 3, the column on the left and the column on the right, which are virtually identical. This occurs because, remember, we've split 1 6-carbon chain into 2 3-carbon chains. And so for every NAD that is reduced and every ATP that is produced on the left-hand side, the same thing is occurring on the right-hand side. Therefore, up to the production of pyruvate, we have 4 ATP and 2 NADH, or 2 reduced NAD. The end product of that 3-carbon chain is pyruvate. The question is, what happens to pyruvate after glycolysis? Well, there are several fates of pyruvate, and we're going to discuss the main 2 on the next couple slides. At the point of pyruvate, glycolysis may proceed in one of 2 different directions. The first is fast glycolysis. The other is slow glycolysis. Fast glycolysis is also known as anaerobic glycolysis. Anaerobic meaning that it can occur when there's not enough oxygen in the cellular environment. When anaerobic glycolysis or fast glycolysis occurs, pyruvate is reduced to lactate or lactic acid. This occurs by the oxidation of the NADH that was originally reduced earlier in the glycolytic pathway. Right after the 6-carbon chain was split into 2 3-carbon chains, we reduced 2 NAD. That NADH, as a result, provides the hydrogen proton to the pyruvate molecule to create lactic acid. Slow glycolysis, or aerobic glycolysis, on the other hand, is when pyruvate is shuttled into the mitochondria for oxidative metabolism. Rather than the NADH providing a proton to the pyruvate molecule, now NADH can also be transported into the mitochondria where it can undergo oxidative phosphorylation. And the proton can be passed down the electron transport chain and even more ATP can be produced. So in summary, glycolysis started with a 6-carbon chain, a 6-carbon carbohydrate, either blood glucose or muscle glycogen. And we end with 2 pyruvate molecules, which are 3-carbon chains. Along the way, if we start with blood glucose, we initially lose 2 ATP, but we gain 4 ATP later on, resulting in a net ATP production of 2. With muscle glycogen, we lose only 1 ATP, we gain our 4 ATP, and we result in a net ATP production of 3. Then when pyruvate is produced, there's one of 2 pathways. It can either be converted into lactic acid in an anaerobic cellular environment if the exercise intensity is high enough, or it can be shuttled into the mitochondria for oxidative metabolism if the exercise intensity is low enough and more endurance in nature. We know that there's 2 directions that pyruvate can go. The question is, how does the body know what to do with pyruvate? Well, it's primarily determined by the work or the energy demand from within the muscle. If the exercise rate is sufficiently high and the intensity is sufficiently high, it will not allow enough time for much oxygen to be present in the cellular environment. So therefore, fast glycolysis will take over and pyruvate will be converted into lactate. But if the intensity of the exercise is low enough and the exercise can continue on, then that will allow time for sufficient oxygen to be present. Then slow glycolysis takes over, and pyruvate is shuttled into the mitochondria where it can go through the Krebs cycle, along with NADH where it can be oxidized and ATP can be produced. An example of a high rate of energy demand would be something like the 1,200-meter sprint or the 1,200-meter run. It's over the 3 minutes that is generally regarded as the primary energy provider by anaerobic glycolysis. However, the intensity is such that fatigue plays a major factor. Ideally, you would want to run the race maximally so that as you cross the finish line, you are completely fatigued. So the intensity is high enough that pyruvate, most of pyruvate is being converted into lactic acid. Whereas, for example, 30 or 40 minutes on the stair climbing machine, where the exercise is primarily endurance related, enough oxygen will be present in order for slow glycolysis to take over and pyruvate will be shuttled into the mitochondria for the Krebs cycle. It may be helpful to take a quick sidestep for the next slide and talk about the differences between lactic acid and lactate. Lactic acid is initially produced as a result of pyruvate being reduced by NADH. When lactic acid is produced as the end product of glycolysis, in other words, anaerobic glycolysis, lactic acid immediately dissociates its proton, and it's the corresponding accumulation of hydrogen protons or hydrogen ions that creates the negative feedback. Lactate, on the other hand, is not believed to be fatigue producing. Lactate is simply a marker in the blood of lactic acid production. So the negative feedback of the hydrogen ion accumulation by lactic acid, by the production of lactic acid, can be things such as calcium released from the sarcoplasmic reticulum. We know that calcium has to be present in order for a muscle contraction to occur. And when we fatigue, hydrogen ions can feed back and affect the release of calcium from the sarcoplasmic reticulum. Actomyosin formation is another. This is maybe somewhat of a new term, actomyosin. Actomyosin just refers to the complex of actin and myosin when they combine. So when the myosin head attaches to actin, it becomes an actomyosin complex. Well, for the myosin head to bind to actin, there's a certain binding affinity. When there's enough hydrogen ions in the cellular environment, it reduces that affinity between actin and myosin. Another negative feedback system that hydrogen ions can feed back and disrupt is the glycolytic enzyme activity. We mentioned earlier phosphofructokinase. Phosphofructokinase, or PFK, can be sensitive to the internal concentration of hydrogen protons. Altogether, how the concentration of hydrogen protons affects the body can result in fatigue, or does result in fatigue. As we exercise at a high enough level, fatigue will ensue, and it's generally related to hydrogen ion accumulation, which also reduces the internal pH of the cell and increases the acidic environment of the cell. Lactate, on the other hand, may not cause these issues. Lactate, again, is not believed to be fatigue producing and just a marker in the blood. So when you read studies or when you hear of markers of lactic acid production, lactate is what's being measured because it's readily available in the blood to measure. Lactate, ironically, also can be an energy substrate. The term gluconeogenesis, or the formation of carbohydrates from a non-carbohydrate source, can occur with lactate. That is, as lactate is produced, or lactic acid being converted to lactate, as lactate is produced, it can be transported to the liver where it can undergo the Cori cycle. The Cori cycle is when lactate enters the liver, is converted into pyruvate, eventually converted into glucose, stored as liver glycogen. After it's stored as liver glycogen, it can be broken down again into blood glucose, transported to the muscle, and used as energy. So the Cori cycle is simply the cycle of lactate being converted to glucose and then back to lactate again after being metabolized. Hopefully that will clear up some of the issues regarding lactic acid and lactate being virtually the same molecule. Really, the difference between the two is just that the cellular environment won't allow lactic acid to exist very long. So once lactic acid is produced, the hydrogen proton dissociates and it becomes lactate. So in review of the glycolytic system, we basically have the energy yield of glycolysis being 2 ATP if we start from blood glucose, or 3 ATP if we start from muscle glycogen. And again, in figure 3, as a quick review, we lose 2 ATP if we start with blood glucose by the priming reactions prior to fructose 1,6-bisphosphate. If we start from muscle glycogen, we do not need to phosphorylate, therefore we only lose 1 ATP, so the net ATP production is 1 higher, or 3 ATP. In addition, with slow glycolysis, that is, if enough oxygen is present in the cellular environment, the 2 NADHs that are produced, or the NADs that are reduced in the glycolytic pathway, can also be transported to the mitochondria along with pyruvate. For every NADH that is transported to the mitochondria, where it undergoes oxidative phosphorylation, there are 3 ATP that are produced. Therefore, the 2 NADH that are transported to the mitochondria result in 6 additional ATP molecules via oxidative phosphorylation. That brings us to the oxidative system. The primary function of the oxidative system is to provide ATP for low-intensity activities, endurance events, as we know in strength and conditioning, long-distance running, cycling, and swimming. Pretty much anything that lasts longer than 3 minutes of continuous activity. The substrates involved in the oxidative system are carbohydrates, fats, and proteins. Although if we are breaking down a lot of proteins for fuel, this would mean that we are in a severe catabolic state, and hopefully this does not account for much of the energy that we are producing. But it can occur, and does. This is different from anaerobic glycolysis, or the glycolytic system, where carbohydrates is the only substrate that can be used to be metabolized. Another thing that is unique about the oxidative system is that, again, it occurs in the mitochondria, whereas the phosphagen system and the glycolytic system occur in the cytoplasm or the sarcoplasm of the muscle cell. The mitochondria is commonly known as the powerhouse of the cell, or in this case, the powerhouse of the muscle cell, produces a great deal of ATP. So again, the oxidative system has a very high capacity for ATP production. It just takes a while to get there. We'll go on to discuss the Krebs cycle, which is figure 4. The oxidative system is made up of the Krebs cycle, the electron transport chain, and beta fat oxidation, which we'll talk about a little bit later. The Krebs cycle, however, goes by a number of different names. First, the Krebs cycle can be called the citric acid cycle, or it can also be called in some biochemistry textbooks the tricarboxylic acid cycle, or the TCA cycle. Either way, it's the same cycle that occurs. And it starts with the shuttle of pyruvate into the mitochondria. Figure 4 is a depiction, again, a basic depiction, although it may be intimidating to start with, is a basic depiction of the Krebs cycle. You can see at the top center of figure 4, pyruvate enters into the mitochondria and is immediately converted into acetyl-CoA. There's about 6 key issues to discuss regarding the Krebs cycle, and the first is the conversion of pyruvate into acetyl-CoA. Pyruvate, if you remember, is a 3-carbon chain. Acetyl-CoA, however, is a 2-carbon chain. If you notice off to the left in that reaction, carbon dioxide is produced. This is interesting because everywhere in the Krebs cycle that a carbon is lost in the chain, it is lost in the form of carbon dioxide. Another interesting thing about the pyruvate conversion into acetyl-CoA is that we reduce one NAD. That NAD is reduced into NADH and can be shuttled into the electron transport system, where it can result in an additional 3 ATP. The second issue of the Krebs cycle that we can discuss is the combination of the 2-carbon acetyl-CoA with the 4-carbon oxaloacetate. When these two combine, it creates a 6-carbon molecule called citrate. Acetyl-CoA is the common start point of the Krebs cycle. So synonymous with glucose 6-phosphate, acetyl-CoA exists from either fatty acids or pyruvate. The third thing is the 6-carbon citrate being recycled into the 4-carbon oxaloacetate. Remember earlier when we talked about the conversion of pyruvate to acetyl-CoA, we lost a carbon. We lost that carbon in the form of carbon dioxide. When we recycle citrate into oxaloacetate, we lose 2 carbons along the way. And you can see on the pathway where carbon dioxide is a byproduct of the Krebs cycle. That brings up an important point. That is, carbon dioxide is the primary byproduct of the Krebs cycle. Also along the way, in the recycling of citrate into oxaloacetate, we reduce 3 separate NADs, we reduce 1 FAD, and we produce 1 ATP through substrate-level phosphorylation. Let's review that very quickly. By reducing NAD, we add a hydrogen, NAD being nicotinamide adenine dinucleotide. We also reduce an FAD. The FAD is flavin adenine dinucleotide, and it is reduced to FADH2. FAD is very similar to NAD and is also transported to the electron transport chain, where it yields 2 ATPs, which we'll cover a little bit later in slides upcoming when we talk about the electron transport chain. Third, the GTP that is produced, you can see between succinyl-CoA and succinate, a GTP is produced. That is guanosine triphosphate. For all purposes, that's really the same or synonymous with a production of an ATP or adenosine triphosphate. And that is 1 ATP molecule that is created via substrate-level phosphorylation. Remember that reducing NAD and FAD in the Krebs cycle will result in ATP, but it results in ATP during the process known as oxidative phosphorylation. The fourth thing of the Krebs cycle that we need to discuss is the step of isocitrate dehydrogenase. Isocitrate dehydrogenase is synonymous with phosphofructokinase in the glycolytic pathway, in that isocitrate dehydrogenase is the rate-limiting step of the Krebs cycle. Therefore, it's sensitive to changes in the internal cellular environment and can be affected by a number of different molecules. We've already commented about carbon dioxide being a byproduct of the Krebs cycle. You may recall, though, during simple physiology or human physiology, that when we exhale, we blow off carbon dioxide from the lungs that originate from the alveoli, which comes from the blood. In respiratory physiology, carbon dioxide is known as the acid of the blood. If you wonder where the carbon dioxide comes from, it's here. As a metabolic byproduct, the carbon dioxide must leave the muscle tissue after being created by the Krebs cycle, transported via the blood, and blown off through the alveoli in the lungs. If not, carbon dioxide can build up, and this is known as respiratory acidosis. The last thing I want to talk about regarding the Krebs cycle is that for every molecule of blood glucose, 2 pyruvate molecules are created. Therefore, the Krebs cycle turns twice for every molecule of blood glucose. Pyruvate enters and is converted into acetyl-CoA. Since there's 2 pyruvates, every ATP or NAD that is produced in the Krebs cycle must be doubled in order to tally the number of ATP that are produced by 1 molecule of blood glucose. As part of the oxidative system, next we're going to discuss the electron transport chain. Figure 5 shows a depiction of the electron transport chain, which is also known as oxidative phosphorylation. NADH and FADH2 are shuttled to the electron transport chain. The electron transport chain is located along the inner mitochondrial membrane. The number of ATP that are produced by NADH and FADH are dictated by where they enter the chain. If you look at figure 5 on the far left-hand side, you can see that NADH initiates the chain. Right after the first ATP is produced, you can see that FADH2 enters the chain at that point. Therefore, NADH results in 3 ATP produced, whereas FADH results in only 2 ATP produced. The process of the electron transport chain occurs by transporting hydrogen protons along the chain. You can see the number of carriers of the protons that exist in the chain: coenzyme Q, cytochrome B, cytochrome C1, cytochrome C, cytochrome A, and cytochrome A3. Along the way, with NADH, 3 ATP are produced, and again, with FADH, 2 ATP are produced. The ATP are produced with an enzyme known as ATP synthase. The next important thing about the electron transport chain is the fact that oxygen is the terminal electron acceptor. If you look at the far right-hand side of figure 5, you can see oxygen accepting the hydrogen protons, and the resultant is water. And it's important to note that water is the primary byproduct of the electron transport chain. So when you consider everything together, carbon dioxide being the primary byproduct of the Krebs cycle, and water being the primary byproduct of the electron transport chain, water and carbon dioxide are the primary byproducts of aerobic metabolism. So now I think we can look at the total energy yield from 1 glucose molecule. We can break it down into 2 basic sections: 1, slow glycolysis, and 2, the Krebs cycle. Slow glycolysis produces 4 ATP from substrate level phosphorylation. Remember, after the 6-carbon chain is split into 2 3-carbon chains, there are 4 ATP produced as a result of substrate level phosphorylation. As a result of oxidative phosphorylation, there are only 2 NAD reduced during slow glycolysis, but this results in 3 ATP each, which is a total of 6 ATP. So with the number of substrate level phosphorylations being 4 and the number of oxidative phosphorylations being 6, the total gross ATP production from slow glycolysis is 10. Looking at the Krebs cycle, we only have 2 ATP produced from substrate level phosphorylation. Remember the GTP that was produced at that place. And then during oxidative phosphorylation, we have 8 NAD reduced and 2 FAD reduced. Now remember, there were only 4 NAD reduced during 1 turn of the Krebs cycle, but it turns twice for every 1 molecule of blood glucose. Therefore, 8 NADs reduced at 3 ATP each results in 24 total ATP. And for the FADs, there are 2 ATP produced there, which results in 4 total ATP. Adding everything together, we get a total gross ATP production of 40 and a total net of 38. Now remember, if we go back to using 1 molecule of blood glucose, we have to subtract 2 for the priming reactions prior to the split of the 6-carbon chain. But if we start with 1 molecule or the breakdown of muscle glycogen, then we only need to subtract 1, which results in a net ATP of 39. Either way, we're very near 40 ATP produced by 1 molecule of G6P. One thing that we didn't discuss when we talked about the Krebs cycle is fat oxidation. Fat oxidation occurs as a result of the breakdown of free fatty acids that enter the mitochondria and undergo what's called beta fat oxidation or beta oxidation. From figure 4, we can see again our depiction of the Krebs cycle. In the upper right-hand corner, you can see where beta fat oxidation is occurring and where it enters into the Krebs cycle. Beta oxidation results in the formation of acetyl-CoA. In figure 4 in the upper right-hand corner, which is a depiction of the Krebs cycle, you can see where beta oxidation provides acetyl-CoA to enter into the Krebs cycle. When acetyl-CoA enters, it's processed just like we discussed before, where we get ATP produced both from substrate level phosphorylation and oxidative phosphorylation. One of the adaptations to aerobic or endurance training is an increase in mitochondrial content within the muscle, or increased mitochondrial density. The resultant is an increased efficiency in fatty acid oxidation or fatty acid metabolism. This spares carbohydrates for use by the central nervous system to avoid central fatigue. Central fatigue is very debilitating during ultra-endurance events and can result in hitting the wall much earlier. For example, in an untrained state, it's much easier to run out of, so to speak, run out of carbohydrates and hit the wall much earlier. This results in a substantial decrease in intracellular pH. Therefore, one of the adaptations to training is a sparing of carbohydrates so that this intracellular pH does not drop as early and we can exercise much longer during the ultra-endurance event. The total energy yield of one triglyceride molecule includes the metabolism of one molecule of glycerol, which is the backbone of the triglyceride, and 3 fatty acids, each 18 carbon chains. The one molecule of glycerol results in 22 ATP being produced, whereas each free fatty acid results in 147 ATP each. Multiplying 147 times 3 gives us 441. The total number of ATP produced from both the glycerol and the free fatty acids are 463. That's an amazing amount of ATP considering that we get approximately 40 ATP from slow glycolysis in the Krebs cycle. When we talk about protein oxidation, we're talking about amino acids that can be broken down and converted to glucose, pyruvate, and various Krebs cycle intermediates. Figure 4 shows a depiction of where these amino acids can enter into the Krebs cycle. At the top of figure 4, amino acids can be converted into pyruvate, oxaloacetate, and fumarate. Amino acids can also be converted into succinyl-CoA and alpha-ketoglutarate. All of those steps in the Krebs cycle can utilize amino acids. However, the more percentage protein used for energy, the more of a catabolic state our body is in. Therefore, if using proper nutrition and training principles, we can avoid using protein for fuel as much as possible. The process of converting amino acids into glucose is the term we've already defined as gluconeogenesis. This is another place at which gluconeogenesis can occur for energy metabolism. Figure 6 provides a very simplified summary of fat, carbohydrates, and protein metabolism. At the top, we have glycogen and glucose. On the right side, we have protein, and on the left side, we have triglycerides. All 3 of these play important roles in energy metabolism. Hopefully, we can maximize the use of glycogen and triglycerides, or fat, for fuel rather than protein. But you can see by this picture where they enter into the metabolic cycles. As we've stated before, there's an inverse relationship between the rate and capacity of ATP resynthesis. Rate refers to how fast ATP can be created, and capacity refers to how much ATP can be created. Table 1 shows a depiction of where those energy systems lie in this ratio between rate and capacity. In Table 1, we see that phosphagen ranks number 1 as far as rate. Remember, 1 is the fastest or greatest rate of ATP resynthesis, but it ranks last, or number 5, in capacity. Fast glycolysis number 2, slow glycolysis number 3, oxidation of carbohydrates, and oxidation of fats and proteins number 5. Keep in mind that as the rate decreases, the capacity increases. Notice that the highest capacity is the oxidation of fats, coming from the amazing number we saw before, 463 ATP produced from the metabolism of one molecule of triglycerides. This versus 1 molecule of blood glucose resulting in about 40. Another concept that we discussed earlier is the extent to which the energy system contributes to the activity. This is determined primarily by intensity and secondarily by duration. Remember, intensity will also dictate the duration of the exercise. Table 2 shows the effects of event duration on primary energy systems used. Notice that on the left-hand column is the duration of the event, the middle column is the intensity of the event, and the right column is the primary energy system used. An important thing to remember when considering the information in Table 2 is that not to oversimplify this information. There are not definite cutoffs. As you can see, where the exercise is intense between 60 and 30 seconds, both the phosphagen and fast glycolysis are the primary energy providers. When we talk about the substrates used in these metabolic processes, we talk about phosphagens, glycogen, glucose, lactate, free fatty acids, and amino acids. Any one of these can be used as substrates. Only the first two, phosphagens and glycogen, can realistically be depleted during exercise. With regard to the depletion of phosphagens and glycogen associated with fatigue, creatine phosphate stores can be decreased about 50 to 70% in the first 5 to 30 seconds of the activity. Creatine phosphate stores can also be virtually depleted or eliminated as a result of high-intensity exercise. ATP stores, on the other hand, can only be decreased to about 60%, even with very intense exercise. Post-exercise repletion or resynthesis of ATP can occur within approximately 3 to 5 minutes after exercise. Creatine phosphate, however, may take up to 8 minutes. Table 3 shows in the far left-hand column the percent of maximal power output. In the next column to the right, primary system stressed. The next column, the typical exercise time, and then the work-to-rest ratios that are recommended for these energy systems. Most post-exercise phosphagen resynthesis is accomplished through aerobic mechanisms. If you look at Table 3, it provides some work-to-rest ratios that allow the time necessary to recover from maximal exercise and allow for the resynthesis of creatine phosphate and ATP. For example, if one were training or using conditioning strategies such as repeated 40-yard dashes, which take approximately 5 seconds, a 1 to 3 work ratio, which was previous recommendations, a 1 to 3 work to rest ratio would only allow approximately 15 seconds of recovery after a 5-second sprint bout, which is a near maximal activity. But according to Table 3, where the phosphagen system is the primary energy provider between 5 to 10 seconds of exercise, we can allow a 1 to 12 or even up to a 1 to 20 work to rest ratio, which will allow greater resynthesis of creatine phosphate and ATP during that time period. Regarding the depletion and repletion of glycogen, it's important to remember that glycogen is stored in the liver and the muscle, and the rate of breakdown of glycogen increases with exercise intensity. That should be fairly straightforward based upon the information we've covered thus far. But recent evidence suggests that one of the training adaptations associated with anaerobic training is a reduction in the concept known as selective glycogen depletion. Selective glycogen depletion can cause early fatigue without total depletion of muscle glycogen, or liver glycogen for that matter. As we become more trained, we can increase the proportional draw of glycogen from all areas, including the liver and all muscles, with exercise. This may decrease the idea of selective glycogen depletion and allow exercise for longer durations. Another concept that has been recently investigated in the literature is the evidence of the rate of glycogen resynthesis is directly related to post-exercise carbohydrate ingestion. If there is inadequate carbohydrate ingestion after exercise, it will impair glycogen resynthesis. But a purposeful increase in carbohydrate ingestion after exercise can actually benefit glycogen resynthesis and allow for better performances in days to come, or even performances in the same day.
This concludes the Bioenergetics of Exercise and Training presentation.